Assalamu alaikum everyone and welcome to my presentation. Let me start by saying a few words about my own background. I am Fatima Asim, student of MPhil second semester Microbiology department. Cause today I am here to present the topic Bacterial Genetic Exchange. Content of my presentation include introduction, mechanism, genetic exchange and antibiotic resistance. Introduction. Bacterial genetic exchange is the process in which the DNA from one bacterium transferred to another bacterium by the means of gene dispersal. Bacteria do not exchange gene by meiosis but commonly exchange small piece of their genome. Bacterial genetic exchange require DNA elements such as plasmid, transposon, integrons, and naked DNA. The genetic exchange has a big effect on bacterial evolution. Mechanism. Three common mechanisms of bacterial gene exchange are conjugation, transduction, and transformation. First, we will discuss about the conjugation. Conjugation is the process by which one bacterium transfers genetic material to another by direct contact. First conjugation was discovered in E. coli K12 strain in 1946. During conjugation, one bacterium serves as the donor while the other serves as recipient. The donor bacterium carries a DNA sequence called the fertility factor and transfers genetic material to the recipient bacterium through conjugation tube. Here you can see the conjugation tube F plasmid, which is also known as fertility factor. F plasmid are episomes that can be inserted into chromosomal DNA. It is first found in bacterium E. coli. The bacterium containing F factor are known as F positive, and those bacterium that do not contain F factor are F negative. When conjugation occurs, the F positive cells act as a donor while the other F negative act as the recipient. In bacterial conjugation process, donor cell attached to a recipient cell with its pilus. The pilus draws the cells together. The cells contact one another. One strand of plasmid DNA transferred to the recipient. The recipient then synthesizes a complementary strand to become an F positive cell. Transduction. In transduction process, the transfer of bacterial DNA from a donor to a recipient bacterium occur through virus particle. A virus particle that infect bacteria known as bacteriophage. Phage attach to a specific bacterial cell surface receptor and inject their DNA. There are two types of transduction. First, generalized. Second, specialized. Generalized transduction, the transduction in which phages transfer any fragment of bacterial DNA from one cell to another cell. It occurs only during lattice cycle. After infection, viral genome may or may not integrate into host chromosome. It means each and every gene has equal chance of getting transferred from one cell to another cell. Generalized transduction occur in variety of bacteria including Staphylococcus, E. coli, Salmonella, and Pseudomonas. Specialized transduction, which is also known as restricted transduction, only restricted genes are transferred from one cell to another. Example, lysogenic phages. These phages integrate their genome in host genome at specific site. During detachment, they take some gene portion of host genome and leave some of their own gene in the host genome. Such phages is called transducing phages. These phages containing host gene infect another host, then it transfers those restricted gene to secondary host. Transformation. In transformation, the incorporation of naked DNA from the extracellular environment. Cells that can be used for transformation are called competent. These cells has the ability to uptake naked DNA from the environment. There are four steps in transformation. First one is development of competence. Second is binding of DNA to the cell surface. Third one is processing and uptake of free DNA. Integration of DNA into chromosome by recombination. Here you can see uptake of DNA by competent bacteria. DNA may be integrated into genome or exist as the plasmid. Genetic exchange and antimicrobial resistance. Horizontal gene transfer is a major mechanism for the growth of antimicrobial resistant. Integron, plasmid, and transposon generally carry resistant gene to more than one antimicrobial, resulting in co-selection for resistance to several antimicrobial at the time. The R plasmid are conjugative plasmid that encodes antimicrobial resistant gene. Thank you so much for your interest and attention.